Welcome, everyone, to another First Sunday Fun Day session with HGP, the Humanists of Greater Portland. My name is Anand Atre, and it is my privilege to be your MC for today's event, which is being recorded. HGP believes strongly in freedom of speech, but the views expressed by myself, our musicians, today's speaker, HGP members, and our guests are not the official views of the Humanists of Greater Portland. For those who are new to humanism or HGP, humanism represents a progressive way of thinking that is informed by science, inspired by art, and motivated by compassion. A humanistic approach to life is grounded in reason and our shared humanity, making a humanistic lifestyle one that aligns with social and planetary responsibility. Humanistic values are not based on appeals to external deities. Instead, they are formed when humans use reason and reflect with empathy on our experiences, collective needs, and capacity for flourishing. Paramount among these values is the commitment to uphold the dignity of all human beings. Before we hear from this week's speaker, I think we have time for another We Sing song. Pete, Paul, and Mike, have you got a song ready for us?
No. We do. Oh, yes.
Oh.
It's not time to make a change. Just sit down and take it easy. You're still young, that's your fault. There's so much you have to know. Find a girl, settle down. If you want, you can marry. Look at me. I am old, but I'm happy. I was once like you are now, and I know that it's not easy to be calm when you found something going on. Take your time, think a lot. Why, think of everything you've got, for you will still be here tomorrow, but your dreams may not. How can I try to explain? 'Cause when I do, it turns away again. It's always been the same old story. From the moment I could talk, I was ordered to listen. Now there's a way, and I know that I have to go away. I know I have to go. No, it's not time to make a change. Just sit back and take it slowly. Now you're still young, that's your fault. There's so much you have to go through. Find a girl, settle down. If you want, you can marry. Look at me, I am old, but I'm happy. All the times that I have cried, oh, that all the things I knew inside. Well, it's hard, but it's harder to ignore it. Go make this if they were right, I'd agree. But it's them, they know, not me. Now there's a way, and I know that I have to go away. I know I have to go.
Wow. Thank you so much to Paul, Pete, and Mike for getting us started with that fantastic rendition of Cat Stevens' Father and Son. It is now my pleasure to introduce today's speaker. Certified genetic counselor specializing in hereditary cancer and precision medicine. She currently practices as a clinical liaison for a diagnostics genomics laboratory. She has lectured for the University of Connecticut's Genetic Counseling Master's Program. She has also served as ancillary faculty for the graduate program at Northwestern University, contributing to their admission selection. In her presentation today, titled The Landscape of Medical Genetics, she will discuss modern clinical applications of predictive and diagnostic genetic testing, the role of genetic counselors, and emerging advancements shaping the future of genomic medicine. It promises to be an extremely educational presentation and one that I'm very much looking forward to. So please join me in giving a big HGP welcome to the marvelous Mary Hickman.
My microphone working? Can we hear me? Wonderful. How lovely and thrilling and terrifying it is to be on this side of the podium today. And such a full house. Well, as I was saying, this is definitely, I think, maybe a lofty topic. Landscape of medical genetics could be an entire semester of a college course. So I think I'm really just going to be kind of surface scratching some of what I do as a medical genetic counselor, what medical genetics really is and what we can look for, what the kind of spectrum of genetic health is. Because I think when we think about our genetics, it's a little bit intangible of what does that mean? What does that actually affect in our healthcare? How much is lifestyle and exposures versus our actual ingrained genetic code? So my hope is that I will kind of pick on a couple of different things, different subjects, and that might inspire a really good Q&A for us to dig more into what might interest you about your genetics. I will say, please do not ask any personal medical questions. I can't give any medical advice today. Okay, all right, so let's start with just a little bit of basic genetics 101. I don't want to dig too much into the biochemistry of genetics because when we think about if you go in to see a healthcare provider, you don't want an entire lesson in how genes work. You want to know, how does this actually apply to my health and what does this information actually mean? But for the purpose of this presentation, our DNA is basically a molecule that codes for our development, our growth, and how our bodies function. It's kind of like the instruction manual for our body's development. We have DNA in every single cell of our body. And it's comprised of a double helix, so a twisted ladder of sugars and phosphates. And the rungs of the ladder are these nucleotide bases. So we designate them A, T, G, C. This is the alphabet soup of our genetics. So if you see a genetic code written out, it's going to be many, many A, T, G, G, C, A, G. I can't read that. If you gave me a genetic code on a book and all those letters listed out, it's gibberish to me. So that isn't necessarily what we're interpreting when we're interpreting genetic data, but that is how it is sequenced and read. So that's the basic building blocks of our genetic code. So this is called the central dogma. And what this states is that our DNA is constantly replicating itself. Every time a cell divides in our bodies, the DNA is copied and the exact same DNA is present in both cells. So we have constant cell turnover, some more than others, like our skin cells constantly, and they share the same DNA every time they replicate. So that DNA transcribes information for RNA, which is kind of like a messenger that then takes that genetic information and translates it into proteins. Proteins or enzymes are kind of like the workhorses in our bodies that actually perform functions throughout. So the reason our bodies work is because we have proteins doing the work. We might be stuck.
If it gets stuck, we can take the cursor down and hit this arrow.
Let's do it that way. Thank you. So in humans, we have tons of DNA. So these are packaged into 23 chromosome pairs. So we have 46 total chromosomes. So our DNA is tightly wound up and packaged and present in the nucleus of our cells. And different cell types express this DNA differently. So as I said, every single cell in our body has the same genetic information. They're all given the same textbook, our brain cells, skin cells, blood cells, but they kind of selectively earmark parts of that textbook to say, this is the information that I actually need to perform my functions. So that's why if we were to do a genetic test on your saliva or on your blood, we could get the same information from it, even though saliva and blood are doing very different things in the body. So how much DNA do we think we share with a banana? Feel free to call out. Oh, that's... I'm hearing 50, 80, 97, all over the board. So for our coding genes, the rough estimate, and this is contested a little bit depending on how you look at it, but about 60%. And the reason for this is that our genetic information is really specific to how cells function. So across organisms, whether they're plants or animals, flora, fauna, it's all the same genetic code that's helping us to have base cellular functions. So between two individual humans on completely different sides of the world, how much shared DNA do you think that we have? Yeah, 99.9, exactly. 99.9. So really, I kind of say the 0.1% are practically just cosmetic differences. What makes us human is beyond our genetics, certainly, but humans are much more alike than they are different. I think if an alien crash landed and looked at all of us, we'd all kind of look the same to them. So that 0.1% obviously has really big implications for our health, because our appearance certainly, but our health, because there can be genetic predispositions that impact our risk for developing certain types of diseases. So the fact that that is isolated to just such a small proportion is amazing, really. So let's talk about the hereditariness of it all. Our genetic information is inherited from our parents. We get half from each. I say that with an asterisk because that's not entirely true. But essentially, we get recombined genetic information from each parent. So we're kind of a mishmash mosaic of our grandparents and our great-grandparents' DNA. We have 2 copies of every single gene, one from each parent. And both those copies usually function together. But sometimes if just one copy is damaged, then a certain disease can be inherited. So that's what we call an autosomal dominant disease. Versus if both copies of a gene are damaged, so we inherit it from each parent, that's a recessive disease. And that's why sometimes a kiddo can get sick and you look back through the family history and everyone has been healthy. It feels like it came out of nowhere. And it's because it might have been a recessive condition that just made its way down through both family trees. So let's talk more about genetic disease and what this means. So mutations, like I said, may be dominant or recessive. What are mutations? They are changes that disrupt the function of our DNA, and they might be present in individual genes. These are like the spelling differences. So I talked about the ATGC. One letter spelling difference in our genetic code can cause a host of health issues, and I will dig into some of those examples that I've seen in clinic. This can also be chromosomal abnormalities. So I talked about how they're packaged into those 23 chromosome pairs. So if we have extra or missing entire chromosomes, that's a lot of information and can have a multi-system effect. So think of things like Down syndrome as an extra chromosome 21. And then epigenetics, which I won't talk too much about because that's its own lecture, but that is changes in how our genes are expressed. So 2 people with an identical genetic code could have different health outcomes because of differences in how that genetic code is expressed. My absolute favorite example of this are imprinting disorders. So this means that, let's say you have a small deletion within your genetic code, but you inherited it from your father. You might be completely fine. It might be benign. But if you inherited it from your mother, it can cause severe developmental delays or developmental issues, organ failure. Really, the scope is broad. But just because of epigenetics, of our genes know which parent they were inherited from, and that changes how they're expressed. So very quickly, I want to touch on the Human Genome Project, because that's not where genetics started, but it's a big one, or the other HGP for this group. So this is the Francis Collins. He was the spearhead of the HGP. It launched in 1990, and fun fact, actually, his wife, Diane Baker, is also a genetic counselor like me. But he was the director of the NHGRI and later the NIH. So at the point where we had started the Human Genome Project in 1990, we already had a really good basic understanding of our genetics. There was some genetic testing available out there. But the question sort of was, what is the entire human genome and what are some of the mysteries in there that if we know something has a genetic cause, we can see it running through families, can we pinpoint the parts of the genetic code that are actually causing it so that we can then test for it? So why sequence the genome? As I said, it helps with opening the door for genome-wide association studies, seeing what diseases are associated with different changes in our genome. It opens the door for broad-based genetic testing for individuals. But what I want to touch on is something that's important in what I do as a laboratory genetic counselor. We do whole genome sequencing for kiddos, for adults, where we take a biological sample, blood sample or saliva, we extract the DNA from the cells within that sample, and we sequence it, and we assemble it. So when we sequence our DNA, it's kind of like we're getting a bunch of little puzzle pieces, and then we have to assemble them all together in the right order. So in order to do that, we need a reference sequence, like the picture on the puzzle box to work off of. We don't want to fly blind here. So the Human Genome Project, the goal was to create a reference sequence, and that will allow us to be able to assemble a genome as we sequence it, identify individual variants that get called by computational models, and then we can potentially report them and figure out why there might be an underlying health concern. So this involved, the Human Genome Project involved sequencing of about 3 billion individual base pairs. So that equates to about 20,000 individual coding genes for proteins in our bodies. If you were to stretch that out in 12-point font end to end, it would be 1,800 miles of letters. It's a lot of the East Coast. So that's a massive amount of data, which means it's going to take a really, really long time for us to sequence that amount of information. So the Human Genome Project started in 1990. It ended in 2003, so it took about 13 years to complete. Another ask the audience, how long do you think it takes now if your doctor were to order you a whole genome test? How long do you think it takes to get a report from that? 3 days. Oh, man, you guys are smart. It can take 2 days. Yeah. That's for really rapid cases and urgent cases, of course. Most cases, it takes maybe a couple months, weeks to months, but we can do it in 2 days. So why do we do it? What do we do it for? How does our medical genetics actually help us? So just talking a little bit about myself and what I do, genetic counselors are allied health providers who have advanced training in genetics and counseling, as the name might suggest. So we take complex family histories, we look for patterns of disease. Sometimes this means patterns of the same phenotype or presentation present through families. Sometimes this means we might see a constellation of symptoms in one single individual that are suggestive of a single underlying genetic change that could affect multiple parts of the body. So we work across specialties. Genetics applies to psychology, ophthalmology, neurology, cardiology, name an ology, genetics is in it. We're also researchers, educators. We work in industry and lab roles. So my role now as a clinical liaison with a laboratory means that I'm educating physicians who are ordering genetic tests who might not be genetics providers so that we can improve access. So I break this down into 3 subspecialties. So genetic information can potentially tell us diagnostic information. So diagnostics refers to finding an underlying cause for an existing health concern. I have done tests that have diagnosed the reason that an old gentleman had eczema. Wasn't really helpful clinically, but it was sort of found incidentally. You have itchy skin and we know why. And I have done genetic tests that found out why a newborn passed away within hours of birth. So the spectrum is wide. Predictive testing, I think, is sometimes of the most interest to people, and this is testing to determine risk to manifest certain diseases. So think sudden cardiac arrest or cancer or neurodegenerative disease like Alzheimer's disease or frontotemporal dementia. So this helps us better understand what preventative measures we might be able to take based on our genetic information to prevent or reduce the risk of the manifestation of disease, or at least to put it off as much as possible. Sometimes there aren't cures. Sometimes there aren't things that we can do with that information, things like ALS or Huntington's. Sometimes it's just helpful to know to make preparatory measures, whether that might be legal measures, financial, getting proximal access to care, just setting up to make sure that your family is supported. So that's kind of where the counseling comes in. Some people want to know this. It's really helpful. Some people don't. And it can be difficult because for entire family systems, the whole family sort of becomes our patient. If we diagnose a condition in one individual, it has broad-reaching implications. And then precision medicine, this is a little bit newer relative to all of genetic testing, but this is analogous to individualized medicine. So think medication or treatment-based testing. An example of this that has exploded since I was even a student. When I was a student, this was all kind of theoretical, but pharmacogenomics. So we have identified certain genes that we can test that can tell us the efficacy of certain medications. So this might be things like psychiatry medication, so SSRIs. Some people respond really well to some and not others, and that can come down to genetic changes. Cardiovascular drugs like warfarin, HIV treatment, opioids and pain medication. And then it's also used in the oncology sphere for chemotherapy or other targeted medications. So I think that that's a really cool kind of emerging section of genetics to help to tailor care and not take a one-size-fits-all model to our health. So I want to touch on the big three for genetic counselors. So there are three subspecialties that we really break down our training into, and I think kind of highlight the spectrum of what types of indications we see, who sees a genetic counselor and why. So pediatric and general genetics is obviously going to be huge and could be its own lecture. So I have really reduced it down here. But here's just a huge array of the types of things that can have genetic causes. So developmental delay and intellectual disability is a big one. Pediatricians now are recommended to order whole genome or exome tests for kiddos with delays. There was a new guideline that just came out last year that pediatricians can order this. Kiddos with autism, that's sort of a hot topic of quote unquote, what causes autism? And in most cases, there isn't an identifiable cause, but when it's profound or syndromic, or we see other health issues, sometimes there is a genetic cause. Skeletal dysplasias, differences in our bone and cartilage development, metabolic disorders, how we metabolize certain substances, and whether or not we have toxic buildup or lack of essential nutrients. Hearing and vision loss. So a single gene can cause both, and progressive, that you might identify in a kiddo who has 20/20 vision and perfect hearing, but we know we can predict that they might have progressive loss of both. Cardiomyopathy and sudden death. If you have a relative who passed away really, really young and sort of unexplained, sometimes we can test other family members and find a cardiogenetic risk, and then we can get them screening or medication to prevent that from happening again. Multiple congenital anomalies is so broad-reaching, but this is birth defects affecting multiple organs. So think like your heart defects, cleft palate, limb reductions, extra or missing fingers and toes. So if a baby is born that has a number of defects, we recommend testing to determine if there might be an underlying reason for that. The key takeaway here, and I could yap about all of these forever, is that a single genetic change can have an entire multi-system impact. So this integrates our care across specialties. So usually someone is coming to see a genetic counselor as a point of care to get a diagnosis and better understand the underlying cause, but also prognosis for what other systems we should be looking at, and then we refer out. So we kind of think ourselves as downstream providers. From an economic standpoint, genetic counselors don't make a lot of revenue for hospitals. We really have to justify our positions, but if we diagnose something that then has a lot of downstream impact, we're referring to a dozen specialists. And that makes hospitals money, which they care about. So let's talk about a quick case example here. The 20-year-old man undergoes a urinalysis as part of his routine annual physical. So pretty standard blood urine test as part of your physical. He's found to have elevated albumin in his urine and trace blood in his urine, which is not normal. So the doctor gives him a call afterwards and reviews it and says, hey, you had some blood in your urine. And the patient says, I've always had blood in my urine. I've had it since I was a kid. That's just me. Who cares? The doctor also notices in his record that he has hearing loss. And well, he knew about it. The gentleman has hearing aids. So he sends him over to a nephrology for a consult, because if we see blood and protein in the urine, that's not great. Could indicate issues with the kidneys. And the nephrologist gets him a renal genetics panel and identifies 2 mutations in COL4A3, which is a collagen gene, and diagnoses him with Alport syndrome. This is not a happy story. This causes risk for end-stage renal disease by age 40. So what can we do about that? ACE inhibitors, angiotensin receptor blockers to maybe delay this renal onset. In some cases, there isn't necessarily a cure, but if we can stay ahead of it, then we can prolong life and hopefully improve outcomes as much as possible. This also may be, he's 20, maybe if he wanted kids, this can tell him, since we know the cause, to test relatives or to test his reproductive partner to see if there might be a risk of passing this down to children. So the downstream effects as well. But this causes hearing loss, ocular abnormalities, and unfortunately renal disease. I'm realizing my examples are a little bit sad, so I'm so sorry, but the profession's a little bit sad. So another example, a newborn undergoes state newborn screening, and newborn screening is present in every single state. The state of Oregon requires it at 24 hours for newborns and then a repeat at 2 weeks. You must apply for a religious exemption if you don't want to undergo it, but newborn screening is really important because it screens for debilitating or deadly diseases that potentially have treatment for us to intervene as early as possible. So kiddo gets a newborn screen, is in the hospital after breastfeeding once, starts having some issues, vomiting, isn't feeding well, gets a little jaundiced, a little yellow, so we're worried about the liver, hypoglycemic. So we're a little concerned here. Newborn screen comes back and finds that there's an enzyme deficiency. So we do some confirmatory genetic testing and find 2 mutations in the GALT gene. So this causes galactosemia. This is a condition that is on the Oregon newborn screen. This causes the inability to metabolize galactose. So they stop breastfeeding, they give them a specialized diet, no galactose, no lactose. We want to prevent liver failure, because if this was not caught, it could have resulted in neonatal demise. So people with galactosemia live into adulthood. There are some impacts. They might have certain developmental delays or intellectual disability, speech problems. It can cause some motor function issues, but better to be an adult with some speech issues and motor issues than to pass at 24 hours. So this is a rare condition, and all genetic conditions are extremely rare. In the Pacific Northwest, 1 in 95,000 individuals is diagnosed with galactosemia. So rare. So now we're going to pivot into prenatal genetics, now that we've given some really sad general genetics examples. Prenatal genetics, it really fascinates me. It's not my specific specialty, but it is kind of like looking into the black box. In all of human history, pregnancy was a mystery until the kiddo came out and we kind of knew what we were working with. We didn't even know the sex of the baby. It was all speculative. Then we got things like ultrasounds and MRIs and other ways to image to take a look to see if there were structural abnormalities or anything else that we should be concerned about. Genetic testing, however, can give us even more insight very early into what we might expect for health outcomes for a pregnancy and whether or not the pregnancy might be viable. So before someone is even pregnant, and I always recommend this if you're thinking about family planning, is carrier screening. So like the galactosemia that I mentioned, these are recessive conditions that could go completely unknown in a family until 2 reproductive partners have a child. So this means that, like I said, there are 2 copies of every gene. So you might have 1 functioning normal copy of a gene and then 1 not working copy of the gene, and you'll be what we call an unaffected carrier, completely healthy. But if you have children and you have children with someone who's a carrier of the same condition, there's a 25% chance, 1 in 4 chance of that child inheriting both copies of the damaged gene and developing a disease. So that still means 75% chance that your child would not have that disease. But this helps us determine risk for rare, disabling, or life-threatening conditions, and it gives us some options. Some people choose to do IVF, where we can actually test the embryos specifically for monogenic diseases. So we can go in at early stages and say, this embryo will have galactosemia, for example, and this embryo will not, and we can select to implant a child without the disease. Some families might choose egg or sperm donation if they feel that they're not compatible genetically to have a biological child with each other. And some people roll the dice and just choose to have a natural pregnancy, 75% chance of everything kind of turning out okay. Scary thought, but more common than you think. These other options are expensive and inaccessible to most people. So we get pregnant. We did carrier screening, everything looked good. We get pregnant. What can we do during pregnancy to test? So this is usually done in the non-invasive realm first, and this involves a blood draw, so absolutely no interaction with the fetus itself. And we test something called cell-free DNA. So this is really cool. This is free circulating strands of DNA from deceased fetal or placental cells that release into the mother's bloodstream. So we can actually do a blood test on the pregnant parent's blood and isolate fetal DNA from that blood. And we can then test that and look for usually large-scale chromosomal differences, so Down syndrome or trisomies. It can also look at the sex chromosomes. So some families call this the gender test. I disagree with its use medically for that, but that is sort of how it's colloquially referred to. It is a screen, it's non-diagnostic. So if we were to see an abnormality for something like this, then we might want to pivot to the diagnostic realm. So that involves invasive procedures, usually amniocentesis is the recommended one. And that is actually a needle that goes and removes some of the amniotic fluid. It, again, does not interact with the fetus itself, but there is an extremely marginal risk for miscarriage because of it, but it's very low. But any risk for miscarriage in a wanted pregnancy is challenging to make that decision. So we test on the amniotic fluid, and we can do much broader testing than non-invasive alternatives. So this is the type of testing we would do if we knew that in that natural pregnancy, a kiddo had a high risk for a known genetic mutation in the family. We could do an amniocentesis, test for that, and tell the family, this child that you're going to have is going to have this disease. After screening, if we see risk for Down syndrome or a more severe trisomy, that Down syndrome, you know, people live long lives, but with a more severe trisomy, extra chromosomes, it could be deadly. So we can get that information as well. And that helps with decision-making. I've had a number of patients say, why would I want to do this if I would never terminate? Because I think that that's sort of the idea here is if we know that a child is going to have a debilitating illness, or we know that it might not be viable, that the decision is to terminate, try again, not let that child be born and go through any suffering. But some people, that's not an option. And genetic counselors are in no way trying to convince someone one way or the other of this, but to just simply provide information. So the way I frame it is, yes, termination is an option, not for some people, but this is also about delivery planning. Maybe that person was going to have a home birth, but we know that this kiddo is going to need tertiary care. So we can prepare for that by bringing them into a medical center that will have the specialists that we know they're going to need. And that way we can make sure that they have a plan, a delivery plan in order to get access to that care. It's also just emotional planning. If we know that a kiddo might have developmental delays, it can prepare families for what comes next instead of being blindsided right after birth. So pivoting into cancer genetics, which is more my bread and butter. All cancer is genetic. So all cancer is caused by random errors when cells multiply. Our cells are constantly dividing and copying, like I said, our DNA. And so all cells have identical DNA. Sometimes mistakes get made. If you've ever photocopied something and you see something messed up on the copy, that's sort of like what happens here. So this Damage can be acquired through replication errors as our cells multiply. It can also be due to carcinogenic environmental exposures. Think things like UV radiation, chemical exposures, tobacco use, cervical cancer with human papillomavirus is another example. So outside exposures that negatively affect our DNA and cause acquired damage. Our cells have certain genes that tell us we're dividing too fast, let's stop. Those are tumor suppressor genes. They also have genes that tell us this cell is sick, let's just die instead. And that's apoptosis. So it's programmed cell death to say this is an unhealthy cell that should not divide in our body. Let's eradicate it and then let the healthy cells proliferate. When we have cancer, it damages those genes. So now cancer cells, which were originally our own cells, are saying, let's divide uncontrollably. And yes, we're sick, but we're never going to die. So that's unregulated cell growth of unhealthy cells. That's a tumor. It doesn't perform the proper functions in the bodies and it impedes our organ function. So all of it is caused by changes in our genes, but only about 10% of cancer is actually hereditary, meaning running through families and inborn. So these are genetic changes that are present from birth. Usually signs that we can see in hereditary cancer syndromes are younger ages of onset that might be typical, rare forms of cancer, multiple cancer diagnoses in one person, or many individuals within a family system that have developed cancer. So these are things that we might look for to indicate if we should do a genetic test to find an underlying hereditary cause. But a lot of people come in to clinic and say, yeah, everyone in my family has gotten lung cancer. Did you smoke? Did you live near a plant? When I practiced in Hawaii, there were some people who came from islands where there was nuclear testing nearby. So there's explanations for why there might be many individuals developing cancer. But if it's sort of unexplained or if they seem related, that's when we can be suspicious of something hereditary. So when we evaluate for hereditary cancers, we take that comprehensive personal and family history. We look to see if there's an indication like I went through. If we do testing and identify a genetic mutation, that helps us determine risk. Your genes aren't going to say you're at high risk for any type of cancer, but one gene might increase risk for breast and ovarian. Another might increase risk for gastric and colon cancer. If we know what the risks are and how high they are, that can inform medical management and preventative measures. So as a case example for this, a 36-year-old woman, healthy, is referred to genetics because of her family history. Her healthcare practitioner went through her family history and said, I'm concerned, let's go to genetics and see what they can do. So we took her comprehensive family history and found out her mom had breast cancer at 45, her mother's mother had breast cancer at 50, and her mother's sister passed away from ovarian cancer at an unspecified age. There's a pattern here. Ages are a little bit younger. 45 is kind of on the young end for breast cancer. Ovarian cancer is a very rare, aggressive disease that has a high chance of being hereditary, certain types of it. So she gets genetic testing and is found to have a BRCA2 mutation. What does this mean for her? Over 60% risk, lifetime risk for breast cancer. And if she gets it, there's also an increased risk for a second diagnosis, elevated risk for ovarian cancer, pancreatic, not listed here, is also skin cancer or prostate. This is a really bad one. There's others that are less severe, but BRCA is a commonly talked about one when we talk about cancer genetics. What do we do? Angelina Jolie effect. She had a BRCA mutation. She had her breasts removed and her ovaries removed. We don't do that immediately. It depends on age and timing. We don't want to affect hormones with an oophorectomy, but that is the best way to reduce risk of developing cancer. If there's a part of your body that has a chance of developing it and we take it out, it reduces the risk. You can't get cancer in an organ you don't have. Again, this is where the counseling comes in for a genetic counselor, because when you find out that information, is that even something that you would have wanted to know and that you feel that you would have acted upon? And for some people, they could go their whole lives happy to never know this. It is also helpful from an insurance standpoint. They love to cover genetic testing like this because it is a lot cheaper to prevent cancer than it is to treat it. So again, thinking about the financials of healthcare, that can be really helpful and is a common question of, is my insurance even going to cover this? So let's talk about some of the future directions of genetic testing. I have just blabbed about all sorts of different types of testing and scenarios and some of the tragedies that can come around this, but we have actually made so many advancements in recent history, and whole genomics is a big part of that. Before, when we did genetic testing, it was just stepwise. We would suspect a single gene, test it, it's negative. Okay, what other gene could it possibly be? It's expensive, it's time consuming, it prolongs the diagnostic odyssey for many people where they have been through the ringer of seeing many specialists and just want an answer. So when we move into the whole genomic sphere, this helps us determine whether or not there's a genetic cause through maybe one or two very quick tests. So it's much more accessible, it's much more cost effective, and it's broader based. So improvements in diagnostics with whole genome sequencing, we actually have a bunch of studies and pilot projects out in the sphere right now that are looking at doing this for newborn screening. We wouldn't report everything in a whole genome for a newborn. There's so many ethical implications with that, but we can essentially mask the genome and target specific portions of it that we want to read through for conditions that we know would affect a newborn. So being selective about what information we would even look at and report in that scenario. We don't want to diagnose Huntington's in a one-day-old, but we might want to diagnose like the galactosemia example I used, something that we have an immediate potential intervention for. Long read sequencing, I won't dig too much into this, but when I had mentioned that when we sequence our DNA, it's kind of like getting a bunch of little puzzle pieces. We're developing long read, and it's used in the research setting now, where you get much bigger puzzle pieces. So it is faster and easier to piece it together and get a whole picture, and it actually improves detection of certain types of mutations that might otherwise not have been caught on a whole genome test. So we're always growing, we're always advancing. I couldn't have even imagined that we'd be at the whole genome level when I was a student, and it happened in a blink of an eye to me. So I suspect long read sequencing is going to be clinically available by the end of this presentation. And then multi-omics. So we talk about assessing our DNA and analyzing that. So multi-omics is looking at the bigger picture. So we see a difference in our DNA, but we don't know what it causes. Can we look at the RNA and see if there's function impaired there? Can we look at the proteins themselves and see if they're absent or if they're misfolded or if they're not performing their functions appropriately? So we're looking at the bigger picture and more downstream. So that's also really big in the wet lab research sphere. That's not mine on the clinical side, but it is really interesting to see presentations like that at conferences. The big one that I know a lot of people have questions about is CRISPR. And I hope that I can answer some of those questions. CRISPR or Cas9, that's what it stands for. I'm not going to go through all that. Essentially, this is genome editing. So it's kind of like a pair of molecular scissors that allow scientists to precisely slice and repair faulty genes. So if we know there's a mutation or damage within a gene, if we can go in and splice it out and replace it with a functioning code, then we could, in theory, cure a genetic disease. So no, we can't change your entire genetic code. I can't, inject you with CRISPR and everything gets changed. We can't use it for designer babies. That's usually a question that comes up. We're not making broad scale edits, but there are certain diseases that we can isolate and that we know affect specific parts of the body. So sickle cell disease, this is huge. This just happened a couple of years ago. So this is caused by mutations in the HBB gene for abnormal hemoglobin. And when someone has sickle cell disease, like this image shows, there's sickling of the actual cells. Blood cells are supposed to be round and bouncy and move through our veins and arteries very easily. If they sickle, then they get caught on each other and it can cause vaso-occlusive crisis. So this is restricted blood flow, it can cause very severe pain and downstream organ damage. We have gene therapy for this. It got FDA approved in 2023. This is the coolest thing. There are 2 FDA-approved therapies that one uses CRISPR to actually silence a different gene that allows our body to produce fetal hemoglobin that does not sickle, and therefore the round blood essentially is created. And fetal hemoglobin is not produced in adults because it has a very high affinity for oxygen. We don't need it. It gets replaced by an adult form of beta globin. But if we can instead introduce fetal hemoglobin, it's sort of like we're just overcorrecting for the issue of the sickling. The other one is a viral vector. So we actually remove blood stem cells from an individual, use that vector to create a healthy copy of the damaged HBV gene, and then put it back into a person, and those blood stem cells replicate and essentially replace the damaged cells. The last thing I want to touch on and then dive into Q&A, I hope I'm good on time, is health equity, which I think should be a topic of conversation around any health conversation around our health, any subspecialty. So our genetic code is almost the same across individuals. There are differences, and how does that affect our health equity? 88%, this was a study in 2021, 88% of participants of large-scale studies of human genetic variation have European ancestry. So when we're studying our genetic code, we are using a very finite population that is not necessarily reflective of all humans. So when we do genetic testing, we can't always tell one-to-one if a change is going to cause disease or not. Sometimes these variants tend to be classified as being of unknown significance. And it's more common in non-European individuals who get tested. So this reduces diagnostic yield for testing for non-European populations, and it reduces access to downstream healthcare. If we can't find a genetic diagnosis, we can't recommend next steps. So how do we solve this? What do we do to fix this? Well, first I'm going to talk about more of the problems, actually. So going back to the Human Genome Project, the reference sequence that we use as the reference for when we do a genome sequence, there have been more since this, but from the get-go, it was only 20 people that actually were used to sequence that entire genetic code. So what we call the fully sequenced human genome is 20 people, mostly European. Some of the cells that we had from those 20 people weren't viable for prolonged research. And there was one gentleman who had very good cells for us to replicate, and the clonal cells had very healthy, clear DNA that we were able to test. So unfortunately, a limitation of this is 70% of the human genome from the Human Genome Project came from a single gentleman in Buffalo, New York. So if you want to ask who the most representative person is of the entire human race, it's some guy in Buffalo. So again, how do we solve this? The human pangenome, so testing of individuals across the board. The goal is to reduce that reference bias of how we analyze the human genome for medical care. If the reference genome represents many individuals, then it helps with interpretation of testing for those individuals. It improves variant discovery and research. We can find new gene disease associations in certain populations. But this does depend on really broad recruitment efforts. So there's a lot of distrust around genetic testing and genetic research. There might be concerns about whether or not there's any perceived benefit to donating that information. There's also cultural or religious reasons people might not give their genetic code. There's certain American indigenous populations who don't believe in doing genetic testing. So it's tricky to get people to come forward and provide that because it's inherently part of us. It is identifying information. We can't fully, truly de-identify genetic code. So there are a lot of concerns around that. But this is an ongoing project that we're looking for, non-white volunteers to provide their genetic information for us to help build this database, essentially. So very exciting things on the forefront, and hopefully will lead to more gene discovery and diagnoses that improve this equity. That's what I have.
I think you covered such a broad range of topics for us within the field of genetics, which was fantastic. What I found particularly illuminating was the case examples, because it showed how the genetic analysis could influence practical steps that people could take. We're going to take our first question from Zoom. It's been typed in, so people aren't following instructions, but that's okay. So you're not going to win the prize, I'll tell you that. But okay, so we'll take the first question from Zoom. Can your genetic analysis show what nutrients are deficient and/or which are needed for health?
Great question, because I did talk about metabolic genetics and our ability to metabolize certain substances. Those are rare conditions. Usually just routine blood testing that is non-genetic can tell us what levels of certain vitamins and nutrients we have. So our genetic code doesn't necessarily give us a roadmap for the best individualized diet, unfortunately. I wish that it did. But that is one of those things that we might be able to see predispositions to certain diseases or deficiencies, but it doesn't tell us actively in that moment whether or not you might have a deficiency.
Great. We'll take question number 1 from the house.
Anecdotally, I've read that some of the tech people in the Bay Area are actually using prenatal screening to try to select sort of what they would consider the smartest and most babies, including like Sam Altman and Elon Musk. That's something that genetic counselors have shied away from in the past. Do you think that as we go forward, there might be legitimate reasons for doing that?
I think that bioethics of that in a clinical medical setting. Listen, I would love to say no. I have been the enemy of progress so many times and been proven wrong. But number one, there isn't single genetic. There aren't a single gene that says this is the intelligence gene. There's not a single gene that says this is the empathy gene, right? So I have not dug into that research, certainly, just maybe out of ethical stand and disinterest in it. But no, we're also have very strong lines about what we will even test in the prenatal setting. So prenatal genome is actually something that is available on that amniotic fluid, as I had talked about, invasive testing. But laboratories can't ethically report everything that we see. So it also isn't a guarantee that certain genetic changes are one-to-one and going to cause a certain outcome. So I think it's certainly an interesting topic and a great ethical conversation, but no, I don't think the utility clinically is there.
We'll go to question number 2 on Zoom, and the question has been typed out. How long has the newborn 24-hour test been required in the US, and how long has it been available?
That's a really good question that I unfortunately do not have the answer to. It has been a long-standing program because in most states, it's not actually a genetic test. It's a biochemical or enzyme test to see if certain things are expressing properly. So if we don't see certain enzymes, then that can, if we work backwards, then that can tell us that there are certain genetic defects. A long time. I believe I even got a newborn screening test when I was born in the state of Maryland, and it's present in all 50 states, but I unfortunately don't have the exact timeline for that. 1960s? 1960s.
Okay, question number 2 at the friendly house.
Thank you. If you have a young adult with a flurry of debilitating conditions and no good answers, so for example, a young woman who can't walk without getting dizzy, so she gets a walker, then she gets a wheelchair, but there's no diagnosis, so insurance doesn't want to provide solutions. Is that a person that should get referred to genetic testing? And if so, who do you ask and what do you tell them to ask for? And what diagnosis codes should they include with their request?
This is branching a little bit into really specific health advice. It can be challenging. If anyone, I talked about the diagnostic odyssey. If anyone has a constellation of symptoms that are otherwise unexplained, unfortunately, genetics is not a silver platter and can't tell us every single thing, but there are some conditions that a specialist might know to look for certain patterns. So any generalist can refer to a geneticist or a genetic counselor, list out the symptoms, and then they can review that and see if it's something that they might have a test available for. Unfortunately, there's many chronic diseases and progressive diseases that we just don't have the answer for, and that's why genetics research is ongoing. But I know a common one that's talked about is Ehlers-Danlos syndrome, which is connective tissue disease that has a whole flurry of health concerns I won't even get into, but Some versions of it have a known genetic cause and some don't. And so genetics clinics get overwhelmed with referrals for it because it's extremely nonspecific and can present differently in many people. But there isn't always a test in order to identify it. So some genetics clinics, unfortunately, don't see people for that condition. So it's dependent, it's variable, but really if you just ask your generalist for a referral to genetics, they'll usually write that, send you off, and the genetic counselor and geneticist can see if any testing might be indicated and go from there. Mm-hmm.
Great. Let's go to question number 3 on the Zoom, and it's another one that's typed out. My insurance companies use genetic testing results to drop coverage. And if so, are there any privacy protections?
Oh, I wanted this question so badly. Thank you. The Genetic Information Non-Discrimination Act, I think 2008. So it survived some bipartisanship. It states, it has 2 sections. Section 1 states that your health insurance cannot discriminate against you based on your genetic code. So if you are found to have a predictive test that says you're at high risk for cancer, your insurance cannot legally raise your rates, drop your coverage, or deny coverage. So there are some exceptions to it. Unfortunately, military individuals with TRICARE don't fall under those protections. And then the second section of it is about employers. So your employers can't demand genetic tests and use that information to make decisions about hiring, firing, or promotions. But I think the exception there is small businesses under 15 people. So there are some limitations to it, but in general, people should not shy away from getting a genetic test. The things that I always tell patients who are getting predictive testing is to consider life insurance, long-term care, or disability insurance, because those are not protected under GINA. So it might be helpful to get those policies in place first before getting a genetic test.
Okay, great. Okay, looks like question number 3 at Friendly House left. So we're going to go to question number
4. Now that 2023, the genetic...
The sickle cell?
Yes. Was bankrupt, that company. That
23andMe, sorry.
23andMe,
Yeah.
I'm sorry, 23andMe went bankrupt. And that information, is that going to be helpful or harmful as genetic code, like professionals go forward? I just am curious how.
So 23andMe lives in a different world than clinical medical genetics. It's a direct-to-consumer test that does not have the same accreditations as a certified medical laboratory, clinical laboratory. Unfortunately, you kind of sign away your rights a little bit when you do a test like that. It doesn't have the same HIPAA protections because your genetic code is your protected health information. So you can't release genetic code to anyone under HIPAA. Same as, your blood tests or any diagnoses you might have, but you do give these third-party companies rights to that information and aggregate data when you send a sample over there. So them going bankrupt doesn't affect the medical genetics of it all. Those weren't databases that we necessarily used, but because they have access to all that data, I think one thing they were trying to do was to sell it to biotech companies for research because they had this whole database of genetic information. So that was a little dicey, and there were a lot of people who were very concerned about where that information was going. But it doesn't affect the clinics of it all. Yeah.
Let's go to question number 5 at the Friendly House.
So with CRISPR, how much material has to be edited and then I guess put back in the person for it to actually affect the person?
Yeah, I'm not a CRISPR expert, so I can give my little 2 cents here. So it's usually just very small sections of the DNA. So when I use sickle cell as an example, sometimes it's just a single letter spelling difference in that genetic code. I talked about how there were billions of these letters in our genetic code, so just one can cause sickle cell. So CRISPR can go in and selectively splice. There's probes that choose sort of end-to-end around that change, remove that, and then splice in a healthy code. So it's not entire large sections of your DNA. Usually they try to minimize the amount so that there aren't potential other downstream effects, or they're not, editing anything else that they shouldn't be. But I don't know all the ins and outs of the technology specifically, but it is very, very small amounts of information that they're going in and editing just where the damage is.
I'm more wondering how much all the person's blood out?
No. So like I said, cells turn over. constantly. So we can't change the genetic code of every cell of your body, because if we injected something into your blood, it's not going to replace your brain cells, right? But because those healthy cells are going to divide and proliferate and turn over, it can actually just be a very small amount that then proliferates and hopefully replaces all of the unhealthy cells.
Like
Yeah.
a little vial,
Like a
whole body.
potentially, yeah.
Well,
Okay, let's go to question number 6 at the Friendly House.
If we were to try to enumerate the types of diabetes according to the data like what you've shown, how many different types of genetically caused diabetes would there be?
There are some already. MODY, so it's a young onset diabetes, has a known genetic cause. Diabetes is tricky because it's potentially polygenic and then also has external triggers as well. So we kind of call these multifactorial diseases. So again, not all health concerns are one-to-one with a specific genetic code causing them. Things like heart disease, diabetes, predisposition for obesity, things like this have lots of other factors and might have multiple genes affecting them.
I remember 2 types, and now I've seen up to 5. But I wonder if you see anything that in the patterns of that matches with any of those kinds of the different kinds.
So there are some that have known genetic causes, yes, but very rarely. The most common types that we see don't have a monogenic cause to our knowledge.
Okay, let's go to question number 7 at the Friendly House.
What do you see as the prospect for CRISPR-related treatment that's being developed for sickle cell and other conditions to be generally available and inexpensive?
Again, I keep thinking I'm like the enemy of progress here and the naysayer, and then things come out 5 years later and my jaw drops. It has to be, it's a vector of a specific code that goes in to cure a specific disease. So it's not a one size fits all. CRISPR, the CRISPR treatment for sickle cell can't be used to cure like macular degeneration or something like that in the eyes, right? So you need to code specific targeted sections of the DNA that we're replacing or that we're hopefully splicing out. So right now, the types of diseases that can be targeted that are being researched are isolated to a certain body system. So that might be something that affects specifically your kidney cells, specifically your skin cells, specifically your blood cells, like with sickle cell. The cost side of it, if we keep getting FDA approval and insurances see the efficacy of it, then hopefully the cost will go down. But these are just very, very specific diseases and isn't broad scale yet, unfortunately. I hope that answers your question. Maybe not.
Yeah, it's very sickle cell, the ones that have been FDA approved, that they're probably extremely expensive.
Probably, yeah. I haven't practiced clinically that got FDA approval, so I haven't seen the patient side of it, of what that looks like. But no, this is a very involved process. All genetic testing, I mean, it's gotten a lot cheaper, but all genetic testing is expensive.
Excellent. Are there any more questions either from the Friendly House or over Zoom? Go ahead, Joyce.
Seems fairly obvious, but when you splice a cell out, must you replace it with a healthy cell? If you didn't, what would the body do?
If you sliced out the section that is damaged, then you've created a large deletion within that gene and it's not going to work anymore. So you made the problem worse, probably.
Wow.
I saw so many hands go up, guys. I'm not a CRISPR expert.
We'll go Jeremy first. Jeremy first.
When's the latest do gene editing? Like, is it prenatal or infancy, or and then you cooked, or?
So like sickle cell has been done in adults, kids and adults, yeah. Technically, yes, we can edit genes in the embryonic stages. Again, that's not something that's clinically available. And I really have to speak from the sphere and my scope of what's medically and clinically done versus research. But in theory, any cell that we can extract DNA from, we can edit that DNA.
you want to go?
Regarding the using CRISPR editing, there are a number of companies now that are trying to do what they call in vivo CRISPR-Cas9 editing. So using viral vectors to infect cells. Right now, for sickle cell anemia, you have to do plasmapheresis. You have to remove blood cells. You have to infect them with the virus. You have to ablate the normal cells from the patient and put them back in to sort of.
So one of the FDA approved ones I talked about does use that viral vector directly in. It's knocking out a different gene. That's why that one was so interesting. It knocks out a gene that.
Well, that's to make the fetal.
To make the fetal hemoglobin. Yep. So that is done.
But is that done in vivo or actually done with ex vivo?
Ex vivo with kids and adults with sickle cell.
Right.
Yeah.
But you can now just inject the virus in vivo and not have to do the plasmapheresis. And there are at least 2 companies that are working on technologies for that type of.
I'd love to see it. I'm excited. Can't wait.
I'll go first, and then we'll go with that. Go ahead.
Taking a view outside of just human genetics, what is your opinion of the idea that there have been diseases, perhaps even COVID-19, that were developed by genetic manipulation?
I really don't have a comment on that. No. Again, outside of my realm, unfortunately.
Go ahead. Yeah, Leslie. Oh, Jeremy again? Well, he already asked his question. Yeah. Okay. Right behind Leslie. Go ahead.
Could you talk a little bit maybe about the relationship between what you do and maybe T-cell therapy? Is there a relationship there?
Not directly in terms of what I do. So the therapeutics, the treatments usually isn't directed from the genetic counselor themselves. So, I mean, in autoimmune and immunology, so the diagnoses can come from genetics, but no, that doesn't necessarily overlap with treatments that we're doing. We're more on the diagnostic side.
Okay, great. Are there any more questions at the Friendly House or over Zoom? Okay.
On AI, how is that having an impact on genetics?
Great question. It is. AI is used in the analysis of the massive amounts of data that we generate from whole genome and whole exome testing. When we use reference sequences, when we use known databases of genetic changes that we've discovered through research, a lot of these data banks exist, and AI can very quickly, faster than humans can, filter through that information, look at the genome that we just sequenced in an individual and flag specific variants that may be of interest. It can also be used to make phenotype-genotype associations. So if someone is sent for a whole genome because they have poor muscle tone and seizures and metabolic acidosis and other concerns, then based on that phenotype, we have databases of what genes we know might be associated with it. And AI is used to go through the sequence and flag those specific genes, and whether or not there are any changes in it that then get interpreted by people much smarter than me for a genetics report. So it is used, and it makes the process a lot faster.
Okay, final ask for questions. Are there any more questions from the Friendly House or over Zoom? Okay. Let's give Mary a warm thanks again for taking the time to spend with us and a fantastic presentation. So it wouldn't quite be an HGP Sunday Fun Day if we didn't end the session with more music. So without further ado, David Buckley and Jeremy Knowles will join Mike and the Barquette Brothers to provide us with more musical entertainment.
All right, we're hopefully getting the lyrics up. Okay. All right. So I'll riff. Keep it going.
Okay. Any more questions?
Yeah. Mary, encore. All CRISPR questions.
That's right.
So this first song is going to be a little bit more related to the actual topic, and then the second song will be a little bit more related to the times.
my mother as my mother left in me? How much of my love will be insane to some degree? What about this feeling that I'm never good enough? Will it wash out in the water? Is it always in the blood? How much of my father am I destined to become? Will I dim the lights inside me just to satisfy someone? Will I let this woman kill me or do away with jealous love? Will it wash out in the water? Is it always in the blood? I can feel the love I want, I can feel the love I need, but it's never gonna come the way I am. I could change it if I wanted. Can I rise above the flood? Will it wash out in the water, or is it always in the blood? How much of my brothers do my brothers wanna be? Does a broken home become another broken family? Or will we be there for each other like nobody ever could? Will it wash out in the water, or is it always in the blood? I can feel the love I want, I can feel the love I need, but it's never gonna come the way I am. Could I change it if I wanted? Can I rise above the flood? Will it wash out in the water, or is it always in the blood? I can feel the love I want, I can feel the love I need, but it's never gonna come the way I am. Could I change it if I wanted? Can I rise above the flood? Will it wash out in the water, or is it always in the blood? Oh, 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 oh,
Had that one in the queue for about a year, but I needed to find a topic that was somewhat related. So thank you, Mary, again. So many ways today. So for this next song, we've sung this one a few times, but for new folks, we want you to be safe out there, but we want you to speak your truth, to go out and make a difference whatever way you can. If you feel like you can't do that safely, or you're not in the mind space, the head space to be able to do that right now, take care of yourself. Take care of the people closest to you, because we're in this for the long haul. This isn't a sprint. This is a marathon. So we need you here for it.
Yelling's getting loud. Keep it down now. Keep it down now. There's talk going round this town. Keep it down now. Keep it down now. Noise is closing in from all sides, warning all the ways to die. They say you better give up, you better give up. I say that's not the right words, my fault. Never give up.
See, never give up, even when we make mistakes.
I'll be an army, no, you're not gonna stop me getting through. I'll sing a marching song and stomp through the halls louder than you. I could surrender, but I'd just be pretending. No, I'd rather be dead than live a lie. Burn the white flag. Burn the white flag. Whisper, move the air. Can you hear me? Can you hear me? Sometimes it's all I've got to spare. Can you hear me? Can you hear me? Noises closing in from all sides, warning all the ways to die. They say you better give up, you better give up. I say I'll never give up, I'll never give up. I'll be an army, no, you're not gonna stop me getting through. I'll sing a marching song and stomp through the halls louder than you. I could surrender, but I'd just be pretending. No, I'd rather be dead than live a lie. Burn the white flag. Burn the white flag. Burn the white flag. Burn the white flag. I'll be an army. I'll be an army. No, you're not gonna stop me getting through. I'll sing a marching song and stomp through the halls louder than you. I could surrender, but I'd just be pretending. No, I'd rather be dead than live a lie. Burn the white flag. 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 Burn the white flag.
Thank you, everybody.
What a prolific performance by the dynamic David Buckley, the jaw-dropping Jeremy Knowles, the mesmerizing Mike Burchett, the pizzazz-filled Paul Barquette, and of course, the phenomenal piano man Pete Barquette. And now that I've succeeded in making alliteration great again, why don't we give our musicians a big round of applause for giving us a wonderful start to the month and giving us a message of hope. We present to you some musical entertainment from the Barquette brothers, Pete and Paul, and Mike Burchett.
Yay. Hey. What are we doing?
Bells.
Oh.
There were bells on the hill, but I never heard them ringing. No, I never heard them at all till there was you. There were birds in the sky, but I never saw them winging. No, I never saw them at all till there was you. And there was music and wonderful roses, they tell me, in sweet fragrant meadows of dawn and dew. And there was love all around, but I never heard it singing. No, I never heard it at all till there was you. was music and wonderful roses, they tell me, in sweet fragrant meadows of dawn and dew. And there was love all around, but I never heard it singing. No, I never heard it at all till there was you. I never heard it at all till there was you. Start off with a purdy one. And now?
And now?
Now.
We don't know.
Oh, I forgot again the ones. Oh, you want to do that now? We're going to do these different father and son songs.
This is Harry Chapin, and he said, it's not about guilt or blame. It's just...
I never thought it was.
Yeah, it's just life.
Life.
A child was born just the other day, came into the world in the usual way, but there were planes to catch and bills to pay. He learned to talk while I was away. He was talking before I knew it, and as he grew, he said, I'm gonna be like you, Dad. You know I'm gonna be like you. And the cat's in the cradle and the silver spoon, little boy blue and the man in the moon. When you're coming home, Dad, I don't know when, but we'll get together then, son. We're gonna have a good time then. Son turned 10 just the other day. Said, thanks for the ball, Dad, come on, let's play. Can you teach me to throw? I said, not today, I've got a lot to do. He said, that's okay. As he walked away, his smile never dimmed. He said, I'm gonna be like him, yeah. You know I'm gonna be like him. And the cat's in the cradle and the silver spoon, little boy blue and the man in the moon. When you're coming home, Dad, I don't know when, but we'll get together then, son. We're gonna have a good time then. My son came from college just the other day. So much like a man, I just had to say, son, I'm proud of you, can you sit for a while? He shook his head and he said with a smile, what I'd really like, Dad, is to borrow the car keys. See you later, can I have them, please? And the cat's in the cradle and the silver spoon, little boy blue and the man in the moon. When you're coming home, son, I don't know when, but we'll have a good time then, Dad. We're gonna have a good time then. I've long since retired. My son's moved away. I called him up just the other day. Said, I'd like to see you if you don't mind. He said, I'd love to, Dad, if I could find the time. You see, the new job's a hassle and the kids have the flu, but it's sure nice talking to you, Dad. It's been sure nice talking to you. And as I hung up the phone, it occurred to me, he'd grown up just like me, yeah. My boy was just like me. And the cat's in the cradle and the silver spoon, Little boy blue and the man in the moon. When you're coming home, son, I don't know when, but we'll have a good time then, Dad. We're gonna have a good time then. Thanks.
There you go, on the run, off again to follow the sun. You're trying so hard to fool everyone who loves you. But don't you know, can't you see that every step you take to be free brings you closer to home as you follow the stars above you?
You don't have to go to the mountain to find it yourself. That magical fountain, the joy of living comes back when the giving is real.
For your broken dreams that no one knows, afraid to scream and ruin the show. You're unable to lean on someone who knows how to love you.
You don't have to go to the mountain to find it yourself. That magical fountain, the joy of living comes back when the giving is real.
Well, the lamp is lit and the fire burns, and all that you give comes back in return. For as long as you live, if you ever could learn that whenever you give, you say without words, I love you. Well, the lamp is lit
And the fire burns, all that you give comes back in return.
for as long as you live, if you ever could learn that whenever you give, you say without words, I love you.
And now?
You want to sing one, Hank? Sure, get up here.
All right.
Round of applause for Hank Robb.
He's gonna. Are you? Wait. We want to sing. We want to sing harmony with you. Hank wanted to do one that he's sure you all know.
Yes, because it's the first Sunday. Why don't we sing something we all know? And I'm betting that you do all know this. There'll be a slight change of lyrics because I do that kind of thing. Otherwise, it'll be perfect. Puff the magic dragon lives by the sea and frolicked in the autumn mist in a land called Honolulu. Little Jackie Paper loved that rascal Puff and brought him strings and sealing wax and other fancy stuff. Together they would travel on a boat with billowed sail. Jackie kept a lookout perched on Puff's gigantic tail. Noble kings and princes would bow where'er they came, and lips would power their flag when Puff roared out his name. Oh, Puff the magic dragon lived by the sea and frolicked in the autumn mist in a land called Honolulu. Puff the magic dragon lived by the sea and frolicked in the autumn mist in a land called Honolulu. Now I had daughters, dragon lives forever, but not so little girls. Painted rings and giant wings give way to heels and pearls. One great night it happened, Jackie Paper came no more, and Puff that mighty dragon, he ceased his fearless roar. His head was bent in sorrow, green scales fell like rain. Puff no longer went to play along the cherry lane. This could not be brave, and Puff that mighty dragon, he slipped into his cave, and now we'll get it right. Puff the magic dragon lived by the sea and frolicked in the autumn mist in a land called Honolulu. Puff the magic dragon lived by the sea and frolicked in the autumn mist in a land called Honolulu.
Round of applause for Hank Robb, folks.
Monday morning. Day is done. Oh, I got it right here. My Pretty Mary.
Oh, the agony of choice for what to play next. Suspicion is killing me here.
This is part of the regular programming.
Good times we had.
Oh, the good times we had.
Another Peter, Paul, and Mary tune. Not quite as recognizable.
How does this start again? I go blank now.
I can't think of it either.
Quick.
Let's do this one we've done for 40 years. How does it go?
YouTube it.
I can't remember the intro.
I can't remember the intro. I can't remember any of it. Good time. God, what a drag it is getting old. The good times we had. Peter, Paul, and Mary.
And I keep getting a different Peter, Paul, and Mary song stuck in my head.
Pretty sure it's in G.
Both of us. Usually it's only one of us at a time. Okay, well,
we'll do a different one, and then we'll think of that one in a minute. Well, we could do the Lightfoot.
Lightfoot. Oh, yeah. What key these days?
I think I do it in D.
No.
Oh, G.
I remember this one. It's called Sit Down, Young Stranger, from the album Sit Down, Young Stranger.
I'm standing in the doorway with my head down in my hands, not knowing where to sit, not knowing where I stand. My father looms above me, and for him there is no rest. My mother's arms enfold me and hold me to her breast. They say you've been out wandering, they say you've traveled far. Sit down, young stranger, and tell us who you are. Well, the room is all gone misty, my thoughts are all in a spin. Sit down, young stranger, and tell us where you've been. Well, I've been out to the mountain, I walked down by the sea. I never questioned no one, no one questioned me. My love was given freely and oft times was returned. I never came to borrow, I only came to learn. At times it did get lonely, but it taught me how to cry. And the laughter came too quickly for life to pass me by. I never had a dollar that I didn't earn with pride, 'cause I had a million daydreams to keep me satisfied. Will you gather daydreams or will you gather wealth? How can you find your fortune if you cannot find yourself? My mother's eyes grow misty, there's a trembling in her hand. Sit down, young stranger, I do not understand. Will you try and tell us that you've been too long at school? That knowledge is not needed, that power does not rule. That war is not the answer, that young men should not die. Sit down, young stranger, I'll wait for your reply. Well, the answer is not easy, for souls are not reborn. To wear the crown of peace, you must wear the crown of thorns. If Jesus had a reason, I'm sure he would not tell. They treated him so badly, how could he wish them well? Now the parlor has gone empty, there's nothing left to say. My father has departed, my mother's gone to pray. And there's rockets in the meadow and ships out on the sea. The answer's in the forest carved upon a tree. John loves Mary, does anyone love me? Oh, yeah. He remembered. Times have changed. All the good times that we've had are gone now. Past this way, only memories will remain. Tomorrow, I thought my dreams would be enough for a while, and all the plans that we made. Hey, we had love, but that was all that we had. Now even that don't seem the same. Peace of mind, where's the happiness we should be sharing? We can't find, no, we can't find any answer in the good times we've had. my dreams would be enough for a while, and all the plans that we made. Hey, we had love, but that was all that we had. Now even that don't seem the same. Peace of mind, peace of mind. Where's the happiness we should be sharing? We can't find, no, we can't find any answer in the good times we've had.